All right, ladies and gentlemen, let's talk about lipids. And lipids are going to be that kind of the odd one of the macromolecules. Okay, lipids do not really exist as polymers. That's why. Okay, it's going to be built still from two or more different types of smaller subunits, but they're not built from these repeating monomers. So if you remember about polysaccharides, how do we make a chain of sugars? We take one monomer, add another monomer, and add another monomer, add another monomer, etc., until you get a huge change of monomers called a polysaccharide, right? What happens when you do, and you will see this in proteins and nucleic acids. For proteins, you're going to have one amino acid, add to another amino acid, add another amino acid. You make a huge chain of amino acids, also called as a polypeptide. Nucleic acids, same thing. You're going to have a monomer called a nucleotide. Add one nucleotide, another nucleotide, another nucleotide, another nucleotide. You make a huge change of nucleotides called a nucleic acid. But that's not really the case with lipids. However, lipids, okay, are mostly made of hydrocarbons. Remember I told you when I was talking about hydrocarbons, I told you that fats have a lot of it, right? Lipids have a lot of these things. And let me just draw you something here really quick. This is going to be an example of a hydrocarbon. I'm going to put dot, dot, dot because it continues. Hydrocarbon means it's a molecule that has carbon, hydrogen, carbon, hydrogen, carbon, hydrogen, right? Hydro for hydrogen, carbon for carbon. So this is just a small little chain that you will see. Get acquainted with this one because you will see a lot of these. Okay. This is a hydrocarbon. And you can continue. Now, hydrocarbons, okay, we say that hydrocarbons are going to be non-polar. That means they're going to be uncharged. Okay, or same thing as saying not charged. They're not charged molecules. Okay, you look at this one over here. Does it have any charges? The answer is no. And the reason why is, let's go back to our bonds. And let's go back to a previous video, right? This, I'm going to do this in a different color. This bond here, okay? This bond over here, okay? The bonds between carbon and hydrogen, the bonds between carbon and carbon, okay? These bonds here, okay, they're going to be non-polar covalent bonds. Non-polar covalent bonds, why? Because the carbon and the hydrogen, they have similar electronegativities, so they're going to be sharing electrons equally. Okay, and that is very important. What about the carbon-carbon atoms bond between those two carbons? Well, it's the same thing. If carbon and hydrogen share similar electronegativities, a carbon bonded to another carbon, well, the same atom, same element, right? So they have the same electronegativities. The point of the fact is because they have similar electronegativities, they're going to be forming a nonpolar covalent bond. A nonpolar covalent bond means that they're going to be sharing electrons equally, therefore, no charge. Okay, so this molecule here has absolutely no charge. This hydrocarbon portion has no charge. And if it has no charge, and I'm going to go back to red, if this molecule has no charges, it's non-polar. If it's non-polar, it's going to be hydrophobic. It does not mix with water. Again, it doesn't mix with water because it is a non-polar molecule. Now, think about this, and this is actually pretty obvious. You probably know the answer. What happens if I put water with oil together? What's going to happen? Are they going to mix? The answer is no. They will not mix. They don't mix. They're not friends. The reason why is because oil is hydrophobic, like water. Why doesn't it like water? Because oils are nonpolar. And because the molecule is nonpolar, has no charge, it cannot hang around with something that has charges. For example, a water molecule. Okay, before we move on, one more thing I have to say about this slide. Three main types of lipids. We'll talk about main types of lipids. These are going to be one, triglycerides, two, phospholipids. Phospholipids are extremely important because these are going to make something called your cell membrane. Okay, and then we will not talk about wax. Just know that wax is a type of lipid, but we'll talk about steroids. And steroids is important because that's what some of your hormones are made of. That's what your testosterone, your estrogen is made of. Steroids, and one of the main steroids is going to be cholesterol. And cholesterol kind of has two purposes. Cholesterol, which you think is actually pretty bad to your health, it's actually not bad. It's actually pretty good. If obviously if you don't eat chicharrón every day, right? So cholesterol is actually pretty good because it's also part of your cell membrane, and also it's kind of a base steroid that is going to lead is you're going to use cholesterol to make sex hormones. So let's start with triglycerides. Or fats. Fats, also known as triglycerides. Why tri? Tri means three, right? I'm going to explain that in a second. What is a triglyceride made of? It's made of a glycerol molecule. This is a glycerol molecule, 3-carbon molecule here. Okay, 3-carbon molecule. And 3 fatty acids. Here's where the tri comes from. Okay, tri. 3 fatty acids. So this, ladies and gentlemen, this is a triglyceride. Okay, it has a glycerol molecule, this one over here, and it has three fatty acids. Okay, this is 1, 2, and 3 fatty acids. Okay, this is another fatty acid. This is how we make fatty acids. Now, this is what I was saying before. We're not really adding a carbon and a carbon and a carbon and a carbon together to make a big molecule. That's not how it works. Okay, that's why this is not really a polymer. We have to add smaller parts to another molecule to make it into a fat, right? And what's going to happen is we're going to be adding 3 fatty acids to this glycerol molecule. So how do we make a triglyceride? Well, we know that if we want to make we want to make bigger molecules, we have to use something called a dehydration reaction, right? Dehydration reaction, we're losing water. But if we're losing water, we're creating a bond. We are sticking things together. So glycerol is going to contain 3 hydroxyl groups. If you look over here, this is our glycerol molecule. This is our hydroxyl group. So what happens in this case, okay, in a dehydration reaction, I'm going to do this in blue, this hydrogen and this OH group from the fatty acid goes away. Therefore, we lose water, right? And we're going to be making a covalent bond. We can say it's this one over here. Fatty acids, okay, are hydrocarbon chains with a carboxyl group. Now let's go to the fatty acid over here. It's a hydrocarbon. That means carbon and hydrogens, right? And if you look at this fatty acid here, you'll see that it's a humongous chain of hydrocarbons. Check this out: carbon, hydrogen, carbon, hydrogen, carbon, hydrogen, carbon, hydrogen, carbon, hydrogen, blah 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 blah. Right? Carbon, hydrogen. Okay. If it's a hydrocarbon, that's pretty much just it. So this part is going to be very nonpolar, right? Very hydrophobic. So you see how these fats are very hydrophobic because they have a lot of hydrocarbon, and they can be huge chains of hydrocarbons. Okay, so a fatty acid is made of hydrocarbons and a carboxyl group, which is this one over here, which we will use part of it to make some water. Okay. Now chains or fatty acids can be the same or different. So let's look at this triglyceride. Okay, let's look over here. Let's look at this triglyceride molecule. There you go, like that. Okay, it has a glycerol molecule. Talked about it already. We are going to go through 3 dehydration reactions. Remember dehydration, get rid of water, make a covalent bond, like I show you over here. So let's go through 3 of those because we need to add 3 fatty acids. So here's one bond, another one, another bond, another bond. Okay, those are 3 covalent bonds formed because of 3 dehydration reactions. Now we have a triglyceride. 1 point, guys, in order for it to be triglyceride, in order for a molecule to be a triglyceride, it needs to have the glycerol molecule and it needs to have 3 fatty acids. Therefore, this one over here is not a triglyceride. Here we are making a triglyceride, okay? But this is a whole triglyceride, and it has to make sense. If it's a triglyceride, it needs to have 3 of something, right? So 3 what? 3 fatty acids, right? 1, 2, 3 fatty acids, and its glycerol molecule. Okay, now let me just go really quick, review really quick the fatty acids. Fatty acids, like we said, they're made of hydrocarbons. They're made of hydrocarbons, okay? And do all fatty acids have the same size? The answer is no. Look at number 1. Number 1 is going to be a fatty acid. This is a fatty acid. Fatty acid number 1. Fatty acid number 1. Okay. Um, it's long, right? It has 1, 2, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15. 15 hydrocarbons. Wow. What about fatty acid number 2? It has 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17. 17 hydrocarbons, right? Can they all be different? Absolutely. Some of them can be shorter. Some of them can be longer. Okay, and some of them, like fatty acid number 3, can have a double bond. Okay, which is gonna do like a little kink, like it's gonna fold. It's gonna have this position. See, the point of the fact, ladies and gentlemen, is the triglyceride can have three fatty acids that look all like this one, or it can have different fatty acids. Like in this example, you have one that is shorter, one that is longer, and the one that it has a little kink because it has a double bond. Really quick, what are the main functions of fats? Well, energy storage. That's the main function, energy storage. But it has to be for long term. Okay? Has way more energy than sugar because it has a lot of hydrocarbons. Okay, and this have a lot of bond. It has a lot of energy in those bonds, in the bonds of the hydrocarbons, and also in the bonds between the carbon and the hydrogens, and the carbons and the carbons. It has a lot of energy in there. Okay, now understand this. This is energy storage, but this is for long term. Okay, I just told you about polysaccharides. Polysaccharides are for short-term energy. Okay, you eat something, you break it down into glucose, you break down polysaccharides into glucose, you use that glucose right away to be able to give you energy. Then what happens if you have an excess amount of glucose? Then you have to store it in the form of glycogen, all right? But this still kind of short term. Then you can use glycogen again. But if you keep building up more, then you end up getting fats, okay, which are going to be a long-term energy storage. And you will see this energy storage in something called adipose tissue, fat tissue. Another function of fats is for cushioning of vital and sensitive areas, like for example, the kidney. The kidney has a lot of adipose tissue for protection purposes. And then finally, some insulation, layers of fat to prevent heat loss. And you get to see this a lot in, for example, organisms that live in really cold temperatures, for example, the seals, right? They have a lot of fat. And the point of the fact, why do they have it? To conserve energy, right? To keep warm during those frigid, in those frigid environments. Okay, so now let's go into what is saturated versus unsaturated. Okay, saturated fats versus unsaturated fats. It has to do with the fatty acids. Now, what is an unsaturated fatty acid? It's when the carbon skeleton has fewer than the maximum number of hydrogens. Okay, in other words, when it has one or more double bonds, that is the key word. So if a fatty acid has double bonds, it is considered unsaturated fatty acid. So what are saturated fatty acids? The opposite. The carbon skeleton has the maximum number of hydrogens. Okay, it has pretty much all single bonds, all of them. All carbon-carbon bonds, all carbon hydrogens are going to be single bonds. Okay, and these are going to be very, very linear, very, very stiff. There's absolutely no bending. Now let me show you with another picture again. Here we go. This, what kind of fat or fatty acid is this? Okay, well, do I see any double bonds? This one, do I see any double bonds? The answer is no, I don't, right? They're all single bonds. Single bonds, single bonds, single bonds, right? How do I know they're single bonds? There's only one line. That's it. Okay, it's pretty easy to distinguish. This one has the maximum number of hydrogens. Okay, and it's going to be very stiff. Okay, let's go over here. What about this one? Is this saturated or unsaturated fatty acid? Well, what is the definition of unsaturated? It has a double bond, one or more, right? Well, this one has a double bond here, which allows the molecule to kind of bend a little bit, right? It bends a little bit, gets this kind of kink in there. So it makes the molecule kind of bend a little bit. And because there's a double bond there, that means we don't have the maximum number of hydrogens. Okay. Now this, why should we worry about this, or why should we care about this? Okay. Well, think about a book. Okay. Think about a book. You close your book, all your pages are gonna look like fatty acids, like this one, right? They're gonna be all they're all easy. So it's gonna be very, very compact, right? It's gonna be able to close down, and it's gonna be very compact. But then what happens if you have a bunch of pages that are all folded? They have like a folded by the middle, or a corner's folded, or everything like that. When you close it, it's gonna close all the way down? No, it's gonna be like get a little puffy, right? It's gonna get like a little it's not it's gonna be spaces, right? It's not gonna close all the way down, right? It's gonna kind of come out a little bit, right? So think of that as double bonds. Think of that as having unsaturated hydrocarbons or fatty acids. Okay, it's gonna make a molecule. If you're thinking about molecules, a molecule that has three fatty acids that look like this, they're gonna be very, very, they're gonna be able to compress. They're able to like all be on top of each other, okay, and stack on each other. What if you have another triglycerides that have this kind of formation, or this kind of fatty acids? They're not gonna stack all the way in, right? There's gonna be like a couple gaps. There's gonna be some spaces, okay, and that makes a whole difference. Okay, so when we're talking about that, animal fats animal fats, okay, are high in saturated fatty acids, okay? So they're easily stacked on each other. And because they can easily stack on each other, they're going to be solid at room temperature. For example, your butter, your manteca, right? And this is an example over here. This is saturated fat. Look, this is what I'm saying. They all are straight lines. they all close and they are able to stack all together. Okay, they're stack all together. Okay, making it solid at room temperature. And this is not great because it can lead to something like atherosclerosis, for example. Okay, can build up plaques in the wall of your blood vessels. And usually when you go to a doctor, they usually, what do they say to you? Use butter, manteca, or use oils, right? Use plant oils. Well, they're gonna tell you use oil, right? Vegetable oil, plant oil, olive oil, extra virgin oil, whatever. But usually they recommend you to have plant and fish fats. These are much better because they're high in unsaturated fatty acids. Unsaturated fatty acids are those that you see over here. They have double bonds in it. And you see how they're not able to stack? Not stacked. Oh, they're stacked. They're not like very compressed. Okay. There's a bunch of gaps over here, for example. There's a little gap over here. There's a little gap over here. So that allows them to be liquid at room temperature. Okay. So know the difference between saturated and unsaturated fats. Okay. If one triglyceride has at least one unsaturated fatty acid, that makes it an unsaturated fat. Okay. In order for it to be saturated fat, okay, all three triglycerides have to be kind of all stacked together. Okay. All should be, or it should have the triglyceride should have all fatty acids have to be saturated in order to be a saturated fat. Okay, steroids. Steroids are going to be made of fused rings. Okay, they're all made of 4 carbon rings. This is a steroid. Cholesterol. They're going to be varying in functional groups attached to the rings. Remember, those functional groups are going to be like these things over here, right? Going to give that molecule a little bit of a different property. One of the main examples about steroids is cholesterol. So cholesterol, ladies and gents, it's a type of steroid. But cholesterol is important, like I said. They're very important. It's a key component of the cell membrane. Why? Because it's going to control its fluidity. And I'll show you an example later on. And also, I know this is going to be the first function of cholesterol. Another function of cholesterol is that it's going to be considered a base steroid. So it's used to produce other steroid hormones, for example, estrogen and testosterone. So if you look at cholesterol over here, okay, you use this to make testosterone and estrogen. Okay, and how different is this cholesterol from, let's say, testosterone? Not really, right? The only thing that changes is pretty much this, right? This, oh, of course, and probably this part over here. Okay, but you see how we use cholesterol here to modify it into testosterone, right? And the same thing can be done about estrogen. So how different is cholesterol from estrogen, right? Well, they have the same hydroxyl group. Has it's pretty much just this part over here, right? That changes. Okay, it's pretty much just this part over here that changes from here, and the rest is pretty much the same. So you see how we build up molecules, especially very important molecules for your body, out of cholesterol. So cholesterol is important. It's good. Then you have something called phospholipids, and make sure I'm gonna draw like 2 stars, that actually 3 stars. That's how important this is, okay? Phospholipids, very important, okay? Why? Because they made up your cell membrane. When we talk about nucleus, when we talk about the cell, we'll talk about cell membrane, okay? But then we'll talk about other organelles like the nucleus, like the endoplasmic reticulum, the Golgi apparatus, like vesicles. Well, all of those organelles are called membrane-bound organelles. That means that they are organelles made of membrane, and these membranes are also going to be made out of phospholipids. Okay, so phospholipids is kind of the main component of membranes, not just plasma membranes, but a lot of different membranes inside of the cell. Now, what phospholipids are made of are this, and really don't overthink it, ladies and gents. Do not overthink this one. It looks very similar, put here, very similar to a triglyceride. I want to make sure I run this correctly. Very similar, not the same, but very similar. Why? It still needs a glycerol molecule. Okay, that's the same as triglyceride. It is triglyceride has two fatty triglyceride has three fatty acids, right? Phospholipids is going to have only two fatty acids, two fatty acid chains. Okay, and this is kind of the big deal thing. Instead of a fatty acid, is going to have something called a phosphate functional group. And this is the really cool thing about this. I told you that triglycerides, that fats, the lipids are very hydrophobic, right? They don't like water because they're nonpolar. When adding a phosphate group, we give this molecule, this phospholipid, some polarity. Okay? Because of this phosphate group, we make this phospholipid something called amphipathic. It's going to have a polar end. Okay, that means it has an evolute charge. So one end of this molecule is going to be hydrophilic. One end of this molecule, the fatty acids, I want to be nonpolar. Okay, and this part is going to be hydrophobic. So we have now a molecule that has kind of two personalities. Okay, let's look at our picture. And I'm going to erase what I just wrote down right now so I can have some space. So please feel free to, at this moment, pause and write these notes. Okay. What's so important about this molecule, and again, we'll spend a couple of minutes here because this is so important. This is a phospholipid. Okay. Again, it's going to have a glycerol part. Okay. Glycerol is going to have two. It's not going to have three. It's going to have only two fatty acid chains. Okay. Which are going to be these ones over here. Okay. 1 and 2. Okay. And it's going to have a phosphate functional group. This phosphate functional group, I'm going to make a little detour over here, is going to be over here. And because it has a bunch of negative charges here with a phosphate, okay, guess what have we done? This is one molecule, ladies and gents. This is one molecule, okay? Phospholipid is one molecule. But this one molecule is going to have this part of the phosphate group. I'm going to do it like this. This phosphate group is going to give this part of the molecule, okay? It's going to make it hydrophilic. It's going to make it polar. This part of the molecule, I'm going to say, can hang out with water, okay? This other part of the molecule is made of a lot of fatty acids, a lot of hydrocarbons. Okay, so it's going to be hydrophobic, non-polar. Okay, this part of the molecule don't like H2O. So what do we have now? Again, we have an amphipathic molecule that has a, I'm going to do this in a different color. That's going to have, look at over here, that has a polar end, non-polar end. And what happens when you put this molecule in water? Is it going to separate? No, it's going to create vesicles. It's going to create something like a vesicle, like a cell, okay, where it's going to have the heads. Okay, these are going to be the heads of the hydrophilic. Let me do this in a different color because I can barely see. So if I can see, you will not be able to see as well. Okay, we're going to have all of these heads. These are the heads of the phospholipids. And can hang with H2O. The other part is going to be hiding away. The tails hide from water. Okay, I hope you're able to see that. I know. It's not very visible. Okay, there you go. These are going to be the tails. Tails hide from water. And that's why if they get together, okay, then it's going to be forming something called a phospholipid bilayer. Why bi? Here's one layer. Here is another layer. The hydrophobic sides, the hydrophobic tails can hang with each other. No big deal. They like each other. But now the hydrophilic area, the hydrophilic heads, they can actually face an environment full of water. So why do phospholipids why are they so amazing? Why do they make your cell membrane? It's because they're able to separate different environments. Okay, and let me just go to my next slide. Show you this. This is what a plasma membrane looks like. Again, just focus right now on the phospholipid bilayer. Don't look at anything else. Look at the phospholipid bilayer. Okay, it's bi because there's two. One layer. Sorry. So first, I don't know. Let's let me do. I got confused here. How to write something in. Um, one layer. They come together because this part, the tails, are going to be hydrophobic. They can hang with it, no big deal. And I'm going to do this in a different color. The heads, let me yet do this in a different color. The heads are going to be hydrophilic. And so what happens now? Now you have a membrane, you have a barrier. Okay, you have a barrier that is going to separate what's outside of the cell with what's inside of the cell. Okay, you just have created a barrier because of these phospholipids.